Hi. Today we're going to be talking about the last step in cellular respiration, which is the electron transport chain and oxidative phosphorylation. Some people have been asking about the titles of the videos. So these match up with the sections in your textbook. However, these last three videos, glycolysis, the Krebs cycle, and the electron transport chain, are all different steps in cellular respiration. So that's important to know. I'm going to go ahead and explain it first with just words, and then towards the end of the video, I'll do a visual explanation for you as well. So if you remember, the main point of the citric acid cycle or the Krebs cycle was to produce our NADA and our FADA2, which are our electron carriers, and they're going to be carrying those electrons to the electron transport chain. At this point in cellular respiration, that's where most of our energy is coming from, is from the NADA and the FADA2. They are really high energy molecules carrying those electrons. And the reason why they're going to be carrying these electrons to the electron transport chain is to power ATP synthesis via oxidative phosphorylation. So remember, our ultimate goal in all of cellular respiration is to produce ATP, but so far we haven't gotten very much. So the electron transport chain happens in the inner mitochondrial membrane known as the cristae. Those are the little folds you see inside of a mitochondria. And most of these components in the chain are proteins, and you don't need to know the specific names of the proteins or the enzymes, but it's important to note that basically what's happening is the electrons are being pulled throughout the chain from protein to protein, and as they're being pulled, they accept and donate electrons, moving back and forth from being reduced to being oxidized. So reduced being gaining electrons, oxidizing meaning losing electrons. And as they're passed down the chain, they experience a drop in free energy, and finally, towards the end of the chain, they're passed to oxygen, which is known as the terminal or final electron acceptor. And once these electrons are passed to oxygen, they're added to other protons and they form water. So the protons, remember, H+, oxygen, O2, and we end up with our H2O. Once again, these electrons get passed by NADA and FADA2. They get taken to the electron transport chain, and they're passed through proteins. It's important to remember, however, that the electron transport chain doesn't actually generate any ATP itself. What we're getting to is the point where the electrons get moved to the oxygen, which the oxygen accepts them, and then this is what's going to power our ATP synthesis in the last step. So the main point of the electron transport chain is actually to break apart the drop in free energy into smaller steps that release energy in a decent, manageable amount. So instead of going from really high potential energy to really low and releasing a whole bunch of energy all at once, it goes step by step in smaller amounts. This is just a picture to kind of show you where things are moving. So our oxidative phosphorylation is going to happen after the Krebs cycle, the intermediate reaction, and glycolysis. And this is, like I said, happening in the cristae, the inner mitochondrial membrane, these folds of the mitochondria that you can see here. So the actual process that produces our ATP after the electron transport chain is known as chemiosmosis. And if you remember back from the past unit, osmosis is the movement of water from high to low concentration. So this word kind of comes from that and reminds me a lot of that, but chemiosmosis is the movement of ions across the mitochondrial membrane, and this is the movement of ions from high to low concentration within the membrane, and a semi-permeable membrane of sorts. And so what's going to happen is through this process, electrons cause these proteins, when they go through the transport chain, the electrons, these proteins, after they pass an electron through, they pump out a hydrogen ion, aka a proton, from the mitochondrial matrix to the intermembrane space. that hydrogen then moves back across the membrane and passes through a protein complex called ATP synthase. And if you remember, anything that ends in an ACE is an enzyme, and because it starts with synth, we know that means to make, so ATP synthase makes ATP. And this process uses the high concentration of hydrogen ions or protons outside of the membrane to drive the making of ATP. And so the use of this proton gradient, the high concentration of hydrogen protons on one side compared to the other side of the membrane, driving the cellular work, that's what our chemiosmosis is. So this is what ATP synthase looks like, and it's a really crazy enzyme because it kind of looks like a motor. You can see here a rotor. It turns around. And so what happens here is we have our high concentration of protons on one side of the mitochondrial membrane, and on the other side we have a low concentration. So kind of like water coming over a wagon wheel of sorts, the flow of hydrogen ions coming in causes this protein to change shape, and that gives it the sign to begin working. This gives us a bond of the inorganic phosphate to ADP. So as the H plus is coming in, that's what gives us the energy to create our ATP from ADP and P. So it's kind of like a wheel that spins. You put the hydrogens in, and then it has the energy to add the inorganic phosphate to the ADP. So this is another picture just so you can kind of see. This is from your textbook. If you think of the ATP synthase like a rotor, the H plus comes in, and that propels the enzyme to speed up the reaction to get the inorganic phosphate attached to the ADP, forming ATP. And the energy to power this reaction, once again, comes from that proton gradient, the high concentration of H pluses on one side of the membrane. And this is what brings us, so the electron transport chain, when those electrons go through, they create that proton gradient. So that's how the electron transport chain relates to chemiosmosis, is that proton gradient is created by the ETC, and then that proton gradient powers the ATP synthesis. So this is going to show you now a visual explanation of what's going on here. So in our electron transport chain, remember we have NADA and FADA2 that carry electrons. And those electrons move in such a way all the way through the chain. As those electrons are moving through the chain, our NADA and FADA2 are reoxidized and become again NAD plus and FAD plus. You can know that they're oxidized because since they end up with a positive charge, that means they have lost an electron because now they're more positive. So oxidation is lost. And in this process, you'll see that as the electrons get passed through the proteins, those hydrogen ions or protons get pumped out. And we'll worry about those in a minute. Those hydrogen ions, the protons, are being pumped to the inner membrane of the mitochondria. Over here, what you're going to see happening is after all of our electrons get passed through, they finally get accepted by oxygen, which combines with other protons coming through, as well as the oxygen itself, and we end up with water. And that's one of the byproducts of cellular respiration is water. Okay. Once again, it's important to note the importance of oxygen here. So that oxygen is the oxygen that you breathe. And the oxygen you breathe is the last electron acceptor in the electron transport chain. That's what the electrons are moving towards. And because oxygen is so electronegative, he's the one that's pulling those electrons towards himself. So it's very important. That's why we have oxygen in our blood. That's why we need oxygen to breathe, because that oxygen is very important in the process of cellular respiration. So now what about these protons? As you remember, for every electron that gets passed through a protein, we begin to accumulate a set of protons up here. And what you'll see is those protons pass through the ATP synthase enzyme, which is a protein that, just like its name sounds, creates ATP. It makes ATP. And the way that that works is our protons pass through, and then you saw the little rotor. It kind of almost like clicks and turns because those protons are being passed through the ATP synthase, and that's the sign for it to change shape, which allows the ADP to bond to the inorganic phosphate, giving us ATP. So this is a closer look at the ETC. And a couple other important things to note is that our NADH and FADH2, once again, they're passing off those electrons, and they move through the electron transport chain. And each time they do that, we get positive charge coming out. These are our protons. And there's no place for these protons to go. They're just kind of stuck in this membrane for now. So this side of the membrane gets a big positive charge, which is going to be the driving force for the next step. Also note, FADH2 comes in a little bit lower compared to the NADH electrons that are carried, so that's why FADH2 produces slightly less energy than the NADH molecules, if you read that. Finally, the protons move through the ATP synthase enzyme, which basically, like I said, you get H plus in, the proton in, ATP out, just like a rotor. And essentially, this entire process takes all of the energy we've stored throughout this whole process, from your food to the pyruvate to the acetyl-CoA, everything we've done, and is now breaking it down into smaller bite-sized pieces. And that's very important because this is kind of akin to, instead of having a huge ball of fire, we're letting out a little bit of energy at a time in the form of ATP. So this